UC Davis scientists focus on some of the tiniest particles because of their impact on the lake's famed clarity. This is part of TERC's long-term data record of change in the Tahoe Basin. Anne is one of the scientists working here in the laboratory.
The particles we are most concerned about are less than 1/100th the thickness of a human hair, about 1 to 2 microns in diameter. Some of these particles are living algae. Others are inorganic in origin and come from erosion. These clay and silt particles may have been brought in by the streams and storm drains that flow into the lake. Recently, we have found ash particles from wildfires. Our particle size analyzer uses a laser beam to measure the concentration and size of the smallest particles in water samples that are collected from different depths in the lake.
These particles are so small, they can stay suspended in the water for many months. Eventually, they bump into other particles and stick together, and these larger particles sink faster and over time will settle to the bottom of the lake. The living algal particles provide details about photosynthesis occurring in the lake. Researcher Stephen analyzes chlorophyll concentrations at different depths in the lake.
Chlorophyll is a pigment contained within algal cells. The algal particles are filtered out of the water, dissolved, and analyzed on a fluorometer. The higher the fluorescence, the greater the concentration of algae. This graph shows a typical summer chlorophyll profile in Lake Tahoe. Notice that the chlorophyll is very low near the surface, but highest at around 180 feet deep. At that depth, there's an ideal balance of light and nutrients to optimally grow algae. Deeper down, light does not penetrate, so photosynthesis cannot take place.
It's also important to know the types of algae that are present. Dr. Lydia is a research scientist who specializes in algae and spends time peering through a microscope to identify different algal groups. At Tahoe, most of the algae are diatoms, but there are also cryptomonads, dinoflagellates, and chrysophytes. The one group we wish to avoid are the cyanobacteria, also known as harmful algal blooms or HABs, which can make the water toxic to humans and animals. In the last few years, these have started showing up at the shallow edges of the lake, and this is something that we are paying very close attention to. Now, light is important for algae, but nutrients play a role too. And can you tell us more?
Algae require the nutrients nitrogen and phosphorus to grow. These nutrients enter the lake along with sediment particles. Lake Tahoe has extremely low nutrient concentrations requiring very sensitive instruments which use light waves for accurate measurements.
Most recently, the lab has been looking at microplastics that are appearing in the lake. Plastic products that are carelessly left on beaches or at parks may eventually end up in the lake where they break up into smaller and smaller pieces that never biodegrade or decompose.
Our field researchers use a mantral net to collect microplastics from the surface of the lake. I separate the plastics in the samples using density separation and organize each piece of microplastics onto a petri dish. Raman spectroscopy is used to confirm the chemical structure of the plastic.
Why do we care? One reason is that Lake Tahoe is our drinking water, and it is some of the purest water on Earth. Next time you're on the beach or in a boat, make sure that everybody around you is properly disposing of all trash, or better yet, make a commitment to stop using single-use plastic.